do we perform statistical tests? I could leave that open to the audience if anyone wants to answer, but I think just in the interest of time, I'll keep going. But always feel free to turn on your mic and just type up if you feel like any of these slides where I have questions you want to answer. I'll try to take my time. So a statistical test provides a mechanism for making decisions about a process or processes. This will help you to interpret your data correctly, and also so that you know if apparent relationships are meaningful or significant, or if they're just due to chance alone. So there is always the possibility that you could have an observation that seems meaningful to you, but could have just been basically a fluke observation. And so this is how we make sure that we don't have any of those, accidental or fluke chances affecting our data. I'm not sure where I'm going here. I guess I'm going to start with LC50s and LD50s for this toxicologist in the room, which are very similar to chronic EC50s. I don't remember starting here, but here we are. Sometimes when you're calculating LC50s or LD50s, you're supposed to plot your data. Usually probit values are plotted against log doses, and the dose corresponding to the 50% probit becomes the LD50 or the IC50 or the EC50 value. Practically, this approach requires linear regression, and you can use the equation to determine the LD50. So you've plotted your log concentration versus the probability, which is the probit. You can convert responses to probit. I can show you how to do that at some point. And then you take your 50% mark and determine what concentration that occurs at. I'm not sure why I started there, honestly. I did not notice that this was the first slide, so I apologize because it's a little bit of a jump into nothing. So let us review. What types of variables have we got? We've got independent variables and dependent variables. Positive controls, negative controls. What other types of variables are there? I feel like that probit thing was in there from last year because someone asked, and now I just messed up the whole flow, so I apologize for that. All right, well, there's other ways to look at variables. One type of variable is called a discrete variable. This can be any one of the variables and control variables that I just described. Discrete variables are variables that have a binomial or nominal response. They can also be ordinal variables. So these are things that are basically, if you were counting them, count data, a yes, no, sort of categorical, or even if it's a nominal, something that's a whole number kind of response. We also have continuous variables. These can be ratios or intervals or anything along the full whole number system. Did I say whole number? Real number system. Sorry, got distracted. So if we're talking about discrete variables, this is information usually that can be categorized into classifications, and the values cannot be subdivided meaningfully.
All right, so these are values that cannot be subdivided meaningfully. So whole numbers and integers only. And they can be numeric, but they don't necessarily have to be. They could be categorical. So it could be like red, blue, green, or yes, no, or low, medium, high. So here are those examples. So like the number of molecules, the number of tumors, the color of a flower, type of infection, taxonomic groupings, locations, population size. I'm sure you guys can think of many other discrete types of variable measurements or measurements you can take that are discrete. That would be a discrete variable. Within that, there is a binomial response type of variable, which is when there's only two possible outcomes. This is a type of discrete variable, but only two possible values. Whereas a nominal response is like a binomial, but with more than two categories. So male or female, bound or unbound, on or off, dead or alive, these would be binomial responses. If you were looking at nominal, you could have, say, herbivore, omnivore, or carnivore, or other types of categories. So another type of variable, or it sort of falls into the discrete category, or ordinal. So here we have ordered categories. The distances between categories is not necessarily known, so it's not discrete in that way. But the data can be transformed so that numerical ordinal values will be replaced by their rank when they're sorted. So often with ordinal stuff, you're thinking about non-parametric testing, which I'm going to talk about later, with ranks. So we're thinking about things like survey responses, like with a Likert or a rating scale, like on a scale of 1 to 10, rank or give your favorite to least favorite, so not 1 to 10, so more like favorite, least favorite. Rank these in order. Your education level, your fitness or condition scales, cancer stages, these are ordinal data, so first, second, third, fourth. Also, you could have dose categories as low, medium, and high as an ordinal. So low comes before medium, medium comes before high. They always have to go in a certain order, so they're ordered. Hence the word ordinal. All right, we also have continuous variables. So in contrast to discrete data, continuous data includes all rational numbers. It is not restricted to a defined separate values. So between any two continuous data values, there may be an infinite number of other data points, and continuous data are always essentially numeric. So good examples: length, mass, concentration, time. What types of variables are important? Or sorry, why is type of variable important? That's the question. Why? So that we can determine what type of mathematical treatment to perform on the data. We also have to make sure that we design our experiments without bias so that they can be effectively tested using a statistical interference test or inference test. And if we don't understand what kind of variable we are observing and what kind of data we're recording, we could accidentally bias our experimental design. And also so that we can understand the limitations of our own work. So what kinds of conclusions and inferences can we make from our data and which kinds are not possible. So I'm not sure what I mean by how do you know. Oh, how do you know what kind of variable you have? I've made a little flow chart here for you to figure that out. So you've got a variable. Is it quantitative or qualitative? You ask yourself this, am I quantifying something or am I describing something in a qualitative way? If it's quantitative, do you have interval data or ratio data? If it's qualitative, are you looking at something nominal that's being scored, nominal or something that's ordinal that's being scored in order? Do you have a discrete or continuous variable when you're looking at your interval and ratio data? Do you have categorical data? And if you sort of move between these questions, you can figure out if you're starting out with nominal data that's still qualitative, or if you're moving from nominal to ordinal up into the continuous categories of interval and ratio data. So these sort of charts sort of help you figure out what kind of data you have and what kind of variables you're working with. So for instance, what type of variable is mass to charge? You can turn on your mics if you want. Mass to charge is a ratio, so it would technically be continuous. Spectral intensity. This one's a little bit confusing. Usually we're doing counts, so it actually is somewhat of a discrete variable because you'll always have a round number. There's no decimal points. But it feels continuous because the range for spectral intensity is so great. Chromatographic peak area. Chromatographic peak area is a continuous variable because areas are measured as ratios technically. Retention time. Time's interesting because sometimes we feel like it is discrete, but it's actually continuous. You can always divide minutes into seconds, seconds into milliseconds, so on and so forth. Number of peptides, that would be a discrete count data because you're just looking at the number. Percent coverage of a protein. So you've got a sequence, you've got a protein and you've got 75% of that protein covered by your data. Is this continuous or discrete? Personally, I think it's a little tricky, but percentages are ratios, so you could put this in the continuous category. Protein score. So this is more like ordinal data. This is scoring, so this would be discrete. And fold change is a ratio, so it is continuous. And you could go on and on. So I use these as examples for people who are working in my group because these are the sorts of data that we're always looking at.
Danina, just so you know, the students are participating big time, but they forgot that you can't see the chat.
Oh, okay. Yeah, I keep saying turn on your mics. So if you guys are writing anything there, I will not be looking at it until I'm done today. So just remember that. Anyone want to turn on your mic and have a discussion now before I move on?
No, I was just saying that spectral intensity was continuous, but then you immediately after clarified that. So
that one is tricky. We still often treat it like continuous data, though. We tend to treat it that way because the range is so great. But because it's a detector that counts the number of ions that hit it, it's count data. It's discrete.
That's
so weird.
True. Yeah, and then Simon and Rita were also saying stuff, but we forgot. But we'll unmute ourselves next time.
Okay. Thanks, Ab, for letting me know. It's okay. You guys don't have to. If you want to just have a discussion in the chat and it doesn't really need to involve me, that's cool. All right. So back to it. This is a sort of mind bender for those of you who are sampling data. So in actual fact, all sample spaces have a discreteness to them, and all observable random variables have discrete distributions. The continuous distribution is a mathematical construction suitable for mathematical treatment, but in practice, not easy to observe. So in fact, when we're actually observing, we almost always are doing a discrete observation. This is a concept by E.J.G. Pittman, who I don't really know well, but I found this online and I puzzled over it for a long time. So thinking about length, it's a measurement. I just said that length is a continuous variable because you could have that as a mathematical treatment. You could have any number of measurements between 1 and 2, for instance. It's infinite. But in reality, you have to use a ruler. And you have your eyes. You're limited by your eyes, or maybe by the resolution of your microscope and whatever scale you've got there. But when we're actually using scales to measure, like a ruler, or even a scale, we're limited by the finest point, decimal point perhaps, that scale can accurately measure, which means that our observations are always limited by a certain discrete space. Does that make sense? So, I mean, like, yes, you could measure 17.5 centimeters, but can you measure 17.5341 with this ruler? No. All right. Something to consider. All right, so back to the real question at hand and not so much head scratchers. Let's talk a little bit more about sample data. So there are those discrete and continuous variables. We can also look at parametric versus non-parametric data. So we're going to sort of take those concepts and turn them into more statistical terminologies and concepts now. So when you're talking about sampled data, it would be parametric if it makes assumptions about a population, whereas non-parametric data does not make any assumptions. Parametric data, sampled data, uses a normal or follows or tends to be distributed in a normal probabilistic distribution, whereas in non-parametric data, the distribution can be arbitrary. There's usually a mean value for central tendency in parametric data. There must be sort of a central tendency that, goes along with that normal distribution. That's sort of the mean. Whereas for non-parametric data, you tend to use the median value for central tendency. And parametric data requires previous knowledge about the population, whereas non-parametric data generally does not require knowledge about a population. So going to what we were just talking about, parametric data tends to be continuous, whereas discrete data would fall into a non-parametric category, although some continuous data would not necessarily be considered parametric, and you may have to treat it like non-parametric data as well. You can draw much more specific conclusions about parametric with parametric testing. So if your data does happen to be considered parametric or you're able to transform it, this will give you the ability to draw more specific conclusions and do more specific tests with your data. But non-parametric tests are really great because they're simple and they're less affected by outliers. So this gives you the opportunity to look at non-conforming or unusually structured data. And in general, when we think about parametric data, again, we're talking about a distribution that tends to look like a curve, whereas non-parametric data can look like random, noisy bar graphs, and we can still analyze it. Okay, I also wish I had structured that slightly different. Are there any questions about the types of sample data and variables before I move on? And I'm actually going to jump into statistics very soon now. Okay. So with there being a few different types of variables and sort of two types of major types of statistical tests, which would be non-parametric and parametric, although there are more, we could kind of then group our science into two kinds of science, where we have descriptive science, where information is collected without a particular question in mind. It's very observational. You're describing what you observe. Whereas we also do hypothesis-driven research. This is where you design a test. You're trying to explain a specific phenomenon. You have a question and a null and an alternate hypothesis. So options for how this question could be answered by your data. And this is what we are taught as being the scientific method in Western science. Good experimental design attempts to disprove the null hypothesis. That's how I was taught this. And the reason why I explain this is because as soon as I started doing omics, I started to get frustrated because everything I knew about the scientific method was not the same, because it turns out I was doing descriptive science and no one ever taught me how to understand, describe, or defend descriptive science. There's a tendency in Western science to dismiss descriptive science as not being as rigorous as hypothesis-driven science. But there are certain scenarios where science cannot be hypothesis-driven, and a perfect example is in astronomy. We don't have as many labs or ways to test what we observe in space. So we just observe it and try to figure it out and model it. And that's sort of the difference. If you're observing and you can't manipulate, you're doing something descriptive, and then you're often doing things like models. Whereas hypothesis-driven tends to be like, we have a predicted outcomes, we check to see what the outcomes are, and then we determine whether those outcomes are significant or not, or if it was just due to chance. So with that, there's two kinds of statistics. So we have descriptive statistics, which describes the data, and inferential statistics. So studies a sample of the same data. So you're inferring the differences between different subpopulations of data. And if you're looking at this and thinking, okay, how does this relate to the variables that Nina just went over? It might be quite obvious, but if it isn't, you can kind of think of descriptive statistics to being more on the discrete side and inferential statistics being more on the continuous side of observations and data. So descriptive statistics tend to involve things like if you're doing univariate analysis and you're doing descriptive statistics, you're usually looking at the distribution of a single variable. So you could be looking at central tendency, dispersion. You're looking at how the data is shaped. That's how I say it. So the shape of the distribution. So you check to see if it's normal. You're checking to see if it's skewed or if it's blocky with kurtosis. And then you can also do bivariate and multivariate analyses. So this is when the sample consists of more than one variable, and descriptive statistics may be used to describe the relationship between pairs or sets of variables. So now you're going to compare groups and figure out if they correlate, or if there's a relationship you can model with a regression, or perhaps you want to know if your hypothesis-driven research and observations behind that are significantly different. You can do t-tests, ANOVAs, and then you can also do things like PCAs, PLSDAs, and LDAs. So I've got all of this in the descriptive statistics category, but some of these may not be exactly things that, I'm trying to group everything for everybody to understand it, but there are exceptions and ways to bend these in either direction. So first thing that most people do when they're doing what we call descriptive statistics, outside of things like mean, median, mode, and how many data points do you have, you tend to look at the distribution of the data. So you might be doing normality tests. This is to figure out if a distribution of the data is actually well modeled by a normal model or distribution function. So some of the tests that people conduct are listed here, and I have a problem saying this one, but D'Agostino's K-squared test, Lilifor's test, the Kolmogorov-Smirnov test, and the Shapiro-Wilk test. I've got this one bolded and asterisk because I think it's probably the most common one that everybody conducts, but it's not the only one. And the red curve is the standard normal distribution. And then what we see here are some different ways that the normal distribution can be shaped when you've got different types of variation in your data. So why is the normal distribution important? It allows us to do descriptive statistical tests, and it is sort of the standard assumption for all of those. So when we do these parametric tests for when we're working with mostly continuous data, we have all of those tests are based on the assumption that the data are normally distributed. If the data does not meet the assumptions of normality, and it is not normally distributed, and you can't find a way to get it there or transform it so that it can go there, then you may have to do different types of statistical tests, specifically what are called non-parametric tests. And while normality is important, it's not a requirement of descriptive testing, but you have to know that so that you can conduct the right type of test. Okay. So this is a set of slides that I just plopped in here and didn't get a chance to sort of alter. But as we were working through this last year, my lab group and I, it came up that we had these really cool violin plots in our software and nobody really understood them. And so I put these like slides together without too much written text. So I'm going to have to explain these. But they're perfect to talk about right now because violin plots are another way to look at your data descriptively, but are useful for so many different reasons. So a lot of people are familiar with things like box whisker plots, where you have, say, the mean and the range and the full outliers sort of represented by your boxes and your whiskers. So that's what you're sort of looking at here with this black bar with the dot in the middle and the whiskers on the end. You can represent the same sort of thing with a violin plot. So the violin plot, and I guess they call it that because of the shape that you sort of get. It sort of looks like it could be instruments. What you're doing here is you're actually plotting the distribution of your data as a curve and then taking it as a mirror image. So this would be where you compare the distributions of different groups. And if the shape of the distribution isn't necessarily normal, you can see it right off the bat, right? Like this almost has a bit of a binomial distribution. There's two modes there. This looks more normal. This is somewhat skewed, and this is just hard to describe. All right, so again, here's the box plot model where you have your median, your quartiles, and your lower and upper values and any other outside points. And a lot of biologists just love representing data like this, and I totally get why. There's a lot of information on a box plot. A violin plot can contain the same information with your median and your quartiles and your upper and lower whiskers, maybe 95% confident interval. But you also get to see the shape of the data, like you said. So the idea here is that you plot the distribution of the data gets plotted in sort of a shape, and you can understand that there's more data here than there is here and less on the whiskers, and that sort of gives you an idea of whether your distribution is normal or not. So here's a standard normal distribution. Here's what a box plot for that might look like, and here's how the violin plot compares. So if you have these beautiful elliptic kind of shapes, you know you're looking at a nice standard normal distribution. So the way to do this is literally you create a histogram, come up with, you center the bars or mirror image them, but centering actually makes more sense, and then rotate it and replace that sort of distribution with a shape or a curve. So you can see what different distributions end up looking like as a violin plot. So if you have a log normal distribution, your box plot's going to look like this, and you may not look at a box plot like that and instantly think, oh, that's a log normal distribution. But when you see it as a violin plot, you might go, oh, that is very bizarre looking indeed. You can also look at bimodal data. And of course, a box plot is not going to capture a bimodal distribution for you at all. You're not even going to know. But the beauty of the violin plot is that you can see that. Okay. So when would you use violin plots? Well, there's some cool ways you can do this. You can put, for instance, let's say you're looking at data from males and females. You could plot them side by side like this, which gives you an instant sort of visual. But one fun thing you can do is actually cut them in half and join them together so that you can see one of your distributions of data on one side and the distribution of a maybe similar group on a Thursday or a Friday, but you can compare males and females. So you can actually make some cool visual comparisons this way. And this is just a really neat animated GIF that I pulled from this website, and you can get more information on violin plots there. But so if your raw data is distributed like this, and then you could make a box plot out of them, your violin plot would then go, and this is just sort of showing you from normal distributions to various different shapes of data. So this gives you an idea of the shape of your data. When you have a discrete variable that you, continuous variable that you measure, and you really understand how it works, this may not be as useful to you, but when you've got a lot of omics data, for instance, a violin plot could be very informative. Okay. So I'm going to move on now, but there's one way to do descriptive statistics. So, you want to know if your data is normal or not. You can conduct normality tests for sure, but as well, if you just want a quick view without having to learn all of the math, you could just try a violin plot of your data. All right, so then what other ways can we analyze data descriptively? Let's say your data has met the Sorry, are we doing descriptive? I kind of lost my point here. But yes, let's say your data has met the assumptions of normality. So let's go on. You can also conduct correlation analyses. So here you're looking at your data and you're comparing two variables, not comparing, you're plotting the results from two variables. So you're in an XY space and you can actually increase the number of variables that you have more than two. It just gets multidimensional and a little bit more challenging to look at and represent. So everyone should be familiar with the concept of correlation, but just a quick visual. If you have a perfect positive correlation, you basically get this sort of straight line looking relationship, and a positive correlation has a slope that is greater than 1 and sort of goes from, if you were to plot it like this with a y and an x traditionally coordinate, it would go from the origin on the bottom left upwards. And then similarly, in contrast, the negative correlation is going to be in the opposite direction, but also look like a straight line. And the actual perfect correlation coefficient would be a 1. So your best correlation is going to have a 1 value, and if it's negative, it'll be -1. If there's no correlation in your data, you're going to have a 0, and the data will be scattered completely randomly with what looks like 0 relationship. And then you can have a high positive or low positive correlation, and that is just literally how much scatter or no scatter there is in your data, but that there's sort of this overall trend that is increasing and similarly decreasing if you have a negative correlation. And this is a reminder that correlation is not causation. One thing might cause the other, but the other might cause the first thing. We don't really know which one is causing which. They could be linked by a third thing that you haven't actually captured in your analysis, and this could be random chance. So there's two ways to do correlation. You can do this with parametric data using the Pearson correlation analysis, and there's actually probably 2 or 3 more now, but the standard one that all of us are familiar with is the Pearson correlation coefficient. There is an equivalent non-parametric test called the Spearman correlation coefficient. So if you have non-parametric data, but you want to know if 2 variables you've measured are correlated, you can try Spearman. And this is a rank correlation. All right, if you want to sort of understand something more, so I should go back here. With correlation, you're usually looking at 2 variables that aren't necessarily controlled. So response variables that you've measured that you haven't necessarily controlled. But let's say one of your variables is controlled and it's one of the variables that you've actually put into your design. So say dose or time or something like that, where you controlled what you were measuring. Well, then it might make more sense to do a regression. So the observed variable or the response variable would go on your y, but your x-axis would be sort of your controlled treatment variable. So this is, in other words, how you can model the dependent variable based on what's known. You can model the response or the independent variables based on the dependent variables that you've controlled. It assumes there's a mathematical relationship between the variables, so there's causality is sort of implied here. So you're assuming that as you increase the dose or as you measure later on in the time frame, that there is going to be a change in your response variable, and it's due to that. So this is used frequently in toxicology. Again, dose response is the example I used. It could also be used to measure things like biochemical activity, receptor binding, perhaps oligonucleotide at silencing or gene silencing, reporter gene assays, etc. And this can also be used as a model to predict where things might go outside of the controlled range that you had used. So trying to figure out if you can predict what might happen 24 hours after the end of your test, or find out what would happen at lower or higher concentrations. We can also use probit regressions. So I started out with the LC-LD50 slide, which should really be over here. Probit regression analysis is a type of binomial regression used to analyze binomial response variables. So if you've ever been trying to do a regression and you had binomial response data, you might be confused as to how to do that. So in toxicology, we're frequently measuring mortality, dead or alive, and this type of analysis is reserved for parametric data. It's reserved for continuous variables in general. And so probit regression helps you if you don't have that kind of data, if you have non-parametric or binomial response variables. So this will transform a sigmoidal dose response curve to a straight line, which can be analyzed by regression then through least squares or maximum likelihood. This was the standard for toxicology before computers and software were used as common tools. So let me show you. So if you have everything in a parametric way as like a percent mortality, you can make a curve like this. But with probit, you would get something that looks more like a straight line. You can also, in a similar way, do a logit or a logistic regression analysis, which is another way of transforming binomial data into a linear response or linear model. And it's very similar to probit. So here you take the log of the odds. So the logistic of the probability of whether this is going to happen or not. And the relationship between the logit and the probit is almost indistinguishable. So the logit is approximately the square root of, pi divided by the square root of 3 times probit. So it's just got a coefficient there that changes it. You can use this, but you can use this for non-parametric data distributions. So on the logit transformation, you tend to have a whole bunch of observed values that are like response variables at 1 and 2. And then you do a transformation to convert these into the odds, and it turns this pretty funky line that's mostly points at the top and bottom into a straight line. I remember I added this because when we were working with Shreya's data last year, she had hatch or not hatch, or malformation or no malformation kind of data. And we were able to try to change the data into this form to see if there were any, and make comparisons between groups to see if there was any changes. Or that was what we were trying to do anyhow. I don't know if it actually worked. All right. So now I'm going to move on to another way that you can look at data in a descriptive statistics sort of way, and that is principal component analysis. So this is just a visual of what that kind of looks like. So here what you're doing in principal component analysis is you are taking an orthogonal transformation to convert the values of your observations, which could be from a set of correlated variables, possibly. So you're looking at more than one variable with your principal component, usually quite a few. You usually want to have, 5 to 10 variables at a minimum when you're doing PCA. And you purposely transform the observations into linearly uncorrelated variables called principal components. So for the number of variables you have, you will create an equal number of principal components. So if you have 5 variables, you'll create 5 principal components. And these are basically like a regression. You can think of it as a linear regression or a linear mathematical representation of your data so that all 5 of those are not correlated. The purpose of this is to describe the variance in the data set. It's used as a tool in exploratory data analyses, and it allows you to visualize the distance and relatedness between 2 populations of data. So let's say you have 2 or 3 populations or many populations where you've made all 5 to 10 or however many measurements. This is how you can see perhaps if those populations are different. It's also a really good technique for identifying outliers. So in principal component analysis, this is another sort of example with some plant data that I pulled from the internet. There are some assumptions. So first of all, the data set, you have to have a data set that could be transformed into linear combinations of variables. There's no guarantee that the directions of the variance will contain good features for discrimination. Your components with larger variance tend to correspond to interesting dynamics. So when you have principal components that sort of hold variables with lower variance, that usually tends to be thought of as noise. Okay. So you can do this with parametric or non-parametric data. Normality is not an assumption. However, the data should be scaled so that all the variables are on the same scale and the effects of outliers are minimized. So if you're going to compare one variable amongst your set of 5 that has a scale of 1 to 1,000,000 and the rest of your variables have a scale of 1 to 10, those additional digits are going to make that variable have a much larger weight on the analysis. So you have to make sure that everything is scaled to a similar proportion as much as possible. And you can use what's called the Hotelling's t-test to determine what the 95% confidence interval is around your data. And if any of your data points fall outside of that confidence interval, you can identify those as outliers. So those are points in your data that have greater variation than the rest of your data set. Another really cool tool that's descriptive and observational, but starts to move more into modeling. It's sort of like you've looked at your PCA and you've looked at the variance in your data and you said, oh, it looks like these 2 groups have similar variance and these 2 are just much different. That's great. Maybe you want to know why. Maybe you want to understand exactly how this could happen. You can start doing modeling and actually force your data into relationships that are more mathematically described so that you can understand which variables perhaps have the largest effects. So one way to do this would be linear discriminant analyses. So here you're basically converting it's similar to making a principal component, an orthogonal transformation of your data, but you're purposely making your components sort of linear regression. And so another good thing about these is it's sort of similar to logit and probit, the way that this gets done. They use continuous independent variables and can also combine or include a categorical variable. So this explicitly attempts to model the difference between classes of data. So you're looking to maximize the differences between, say, 2 groups but maybe the variables you've measured appear to overlap. So this gives you that extra dimension that you might need. The assumption is that the data is parametric in this case, because you're doing basically a linear regression. So it's similar to linear regression in that sense, where PCA could sort of be like the multidimensional correlation analysis, and LDA would be like the parametric linear regression test. And you have to know which categories and groups you have. So this would be for a more controlled, hypothesis-driven approach, perhaps, but you can also use this on data that you've done more observational analyses with as well. But you must know if there is possibly a nuisance variable that you're unaware of that could throw the data off, this may not be the best approach. So just sort of to summarize again visually what I just said, PCA will maximize the variance between the two component axes, whereas LDA purposely maximizes the components for class separation. So this should give you, it's intentionally trying to find the differences between your different groups. And PCA is not trying to do that at all. It's just trying to maximize the variance that you can plot your data and see how much variation is present. So both find orthogonal linear combinations and make new axes out of your data, and then you plot it to sort of best explain the data. But again, PCA doesn't take into account any difference in class. So you're not modeling, you're not forcing your data into a specific category. LDA maximizes. So I've got about 10 minutes left. I'm going to see what I can get done in the next 5, and then I'll stop and ask questions and wrap up. Partial least squares discriminant analysis, or PLSDA, and I should go back and just say PCA is commonly used in omics analyses. We do this often to look at the variance and identify outliers. And I say LDA and RDAs and these types of things are much more common in, say, ecological studies. And then PLSDA is used a lot in omics as well, particularly more so for metabolomics data, although we've been using it for our proteomics data. And so this is sort of the next level of LDA and PCA. It's a supervised classification method where, again, you're looking for linear regression models where you're projecting the variables and coming up with new axes or components, and you can project your observations into that new space. And the dependent variable is categorical. It is used heavily for bioinformatics and omics, like I said. So if you want to sort of look at this, PCA is looking at the variability. PLSDA is sort of like an LDA where you're doing the same sort of orthogonal projections as PCA, but purposely with each class. So you're trying to model your groups intentionally into this new dimensional space. And then you can actually do something that's called an OPLSDA, or orthogonal partial least squares discriminant analysis, that then takes your variance and purposely makes it so that they are in orthogonal directions. So it transforms one of your groups into another space so that they're even more separated. So these would make sure that there's no correlation between the different groups. This is great for diagnostics and easily interpreted visualization, but you are really forcing a model onto your data here. So you have to be very careful that you do not overfit your data. It does not improve the predictivity of the PLSDA. So in fact, if your model from PLSDA is predictive and gives you sort of a strong predictive outcome, which you can do with tests after the fact. There's permutation tests that give you an idea of how predictive your model is. If you do the OPLSDA, this should not improve your predictability at all, but it does look better. And it may give you variables it may identify variables that more strongly affect the data. Okay. So The difference here between supervised models and unsupervised models are whether you're implementing a classification. So in supervised regression, you're purposely classifying your data and then trying to regress each class. So this is what PLSDA and OPLSDA and LDA do. In unsupervised regression models, you're doing things that are not dependent on class. So this would be regression, logit, probit, and also PCA.